grace period, I guess. 2:03. We'll wait until 2:05 at least. We just start. Okay, welcome. Welcome to the SkIN Canada webinar, one of the last ones for this year. And my name is Annie Phillip. I'm a professor at McGill University, Department of Surgery and Department of Medicine. I work in the field of SkIN, and I'm also the co-director for SkIN Canada, and also a co-chair for the training for SkIN Canada Training Committee. And for those of you who don't know what SkIN Canada is, it is an acronym for Skin Investigation Network of Canada. That is a CHR-funded network with members from all across Canada. And so we are now, this particular webinar is sponsored by the Training Committee of SkIN Canada. And thank you to all the training committee members and SkIN, and also Ken, who has coordinated this effort. And SkIN Canada has a big, vibrant training component, and the training committee is part of that. So I just wanted to acknowledge all the training committee members. These members come from all parts of Canada. We have about 4 trainees representing the various regions of Canada. And we have always a shortage and from the Western provinces, but surplus from Ontario and Quebec, mostly Quebec. So we would like to have more members. If anyone is listening from Western Canada, that would be wonderful. Our theme today for the webinar is advancement of bioprinting and bioengineering and their potential applications to the skin. We have 4 excellent speakers lined up, and we will hold the question answer to the end when there is a panel discussion. So please note down your questions until that panel discussion, which we hope you would all actively participate. We would like to make it as interactive as possible. The only other thing I want to mention is, okay, you can, of course, use chat function and note your questions there as well. I just want to encourage all of you to participate actively during the panel discussion. So we'll move on to our first speaker, who is Dr. Derek Rosenzweig, pardon me, Dr. Derek Rosenzweig. He is an associate professor at McGill University, Department of Surgery, and he is a staff scientist and independent investigator at the Research Institute of the McGill University Health Center, or we call it MUHC. He is an FRQS Junior 2 scholar, and he obtained his PhD from the Miami Miller School of Medicine in cellular and molecular pharmacology. He has a strong background in molecular pharmacology and cartilage and spine biology. And he specializes in biomaterials and biofluidics, which he will talk to us about today, along with the bioprinting techniques. His lab focuses on biofabrication and bioengineering technologies to study musculoskeletal repair and regeneration in the context of bone and spine metastasis. Dr. Rosenzweig will tell us today about his specific technology that he is focused on in his lab for disease modeling and potential applications to skin modeling and skin regeneration. Okay, Dr. Rosenzweig.
Okay, thank you, Dr. Phillip, for that nice introduction. Get this open here. Share screen. This one. Okay, so hopefully everybody can see that. Let's wait for it to reach the, there we go. Okay, so as Dr. Philip mentioned in the introduction, I'm going to give a little bit of an overview today on 3D printing and bioprinting, and how these tools are used in biofabrication of human tissues so that they can be used for one of two applications, either modeling some kind of human disease, which then can be used for therapeutic screening or understanding molecular mechanisms of disease development, or for use in potential regenerative medicine. So can we actually tissue engineer some kind of substrate that can be then used for human tissue engineering applications? So a focus of my lab, like we just said, is utilizing these kinds of tools. Can you guys see my mouse? Yes? No? See the mouse moving?
Yeah,
Okay, perfect.
Laser pointer on the bottom, I guess.
This one
Bottom
here.
left. No, bottom left. Yeah.
This one here.
Yeah.
That's the one. Recorded. Laser pointer. Got it. Perfect. So we're utilizing these kinds of technologies, which is 3D printing and 3D bioprinting, which can then be used to biofabricate some kind of tissue. And in my lab, we're interested in using those biofabricated tissues for screening novel therapeutics or drug delivery devices like nanoparticles. We're interested in regenerative medicine, which you may hear from Dr. Gowrie after for ligament regeneration. We're interested in modeling tumor on a chip in a bone microenvironment, and that's where bioprinting really comes in, because you can spatially arrange cells precisely where you want them and position them in layers next to other types of cells. You can even create channels where fluid flow can occur. So you can really model high-level tissue in its complete form. We're also interested in various types of polyester polymers mixed with bioactive materials like ceramics, which can then be used for bone regeneration or modeling bone cancers. And then we're leveraging these kinds of tools, both of these kinds of tools, to study new types of medicines, like plasma, cold atmospheric plasma medicine, and its ability to promote regeneration or block cancer. So just to give you guys a little overview, I'm not sure how many people know about 3D printing here. Well, the official term for 3D printing is additive manufacturing. And as it suggests, it's additive in nature, meaning that you build up a model, a physical model, from a digital model that you prepare in some kind of CAD software, and you create those physical models in a layer-by-layer format. It's a cost-effective method, and the most important aspect is that you can produce geometries that you cannot produce using casting or molding or CNC milling. So using standard manufacturing principles, you cannot produce something, for example, like this coin trap, which if I can show you, right? So in this coin trap that I'm showing you on the camera right here, the only way that you can't do this with standard manufacturing. You cannot get that coin inside this model. But because this was printed in a layer by layer format, we're able to drop the coin inside and then complete this model after the coin was dropped in there, and you have this interesting coin trap. So you cannot do those geometries using standard manufacturing procedures. Okay, the other interesting thing about additive manufacturing and 3D printing, it has a host of applications in orthopedics, for scaffolds, guided bone repair, surgical tools that can be sterilized and single use. You can use different types of materials. You can use metal, you can use ceramic, you can use plastic, you can use rubber, you can even use biomaterials to 3D print. Okay. So again, what is 3D printing? You go from a CAD design model, right? You draw this in a 3D CAD model, this cup, for example. You then run it through a specialized software called a slicing software, which then converts that model into the individual layers that the printer will make. It's put into a special code called a G code. That G code is understood by the 3D printer, where it can then go and position each one of those layers by extruding some kind of material in this case. There's a lot of different flavors of 3D printing. We have droplet-based printing. You have laser-assisted printing. You have fused deposition modeling. You have this light-based DLP printing, where you have either a liquid resin vat that is then photopolymerized. You have selective laser sintering. And then you have, of course, bioprinting, where we're printing biological materials in some sort of extracellular matrix that's printable. Okay, so the other aspect of additive manufacturing is that it can also be known as bioprinting. And this bioprinting takes form in biofabrication. And that is when cells are integrated or overlaid on top of some kind of substrate, and you begin to generate a tissue-like structure with some kind of matching function. So this is an example here I took from a paper that you can just see. You can build replicas of ears, different types of materials that would represent bone or cartilage. And there's several different kinds of materials that can go into this, from plastic polymers to metals to ceramics to all these different naturally derived materials like hyaluronic acid, chitosan, alginate, silk, elastin, fibrin, down to these synthetic ones like pluronic acid, polyethylene glycol, gelatin methacrylate, or methyl cellulose. So any of these could be used as materials for biofabricating living tissue-like substrates. So why is 3D printing and biofabrication interesting? And how did, for example, how did I even get into this? Well, I can share with you a really quick story that sparked my interest and really got me to deep dive into this area. So what you're looking at here is one figure from a study from Dietmar Hutmacher's group out in Australia. And what they did several years ago, I think this work started in the mid 2000s, or around 2005, 2004, he started doing this, is he was 3D printing polycaprolactone, which is a biodegradable polyester, and 3D printing it into a bone structure that would fit into a segmental defect in a sheep. And he filled this biomaterial with a powder mix of bone morphogenic protein, and implanted along this segmental defect, which you just see right here, implanted this biofabricated scaffold, put a fixator over it, and then he shows a video in his presentation, which is just amazing. 12 months later, they removed that metal fixator, and the animal's bone is completely healed, completely grown through and replaced with natural bone, and the animal is running around on that leg where they fixed that full segmental defect. So you fast forward a few more years after that study was done, and interesting enough that the clinical group that works with Dietmar Hutmacher, they approached a patient that actually had a very bad bone infection in his lower leg and was faced with the potential amputation. And this 27-year-old man was presented with an opportunity to put in this polycaprolactone scaffold to help regrow bone over a metal rod implant. And what you can see here is the clinicians showing him exactly how it would be done. This metal implant would be put across the lower leg, and then this scaffold that is actually a CT scan replica of this side of his limb was then placed over with that polycaprolactone material, placed in, and actually acted as a very slow guide to allow new bone to grow in across that plate. 2 years after that, so that was 2017, 2 years after that, 2019, this man still has his leg, did not have to have it amputated, and is able to walk around on that limb now. So I thought this is fantastic, and I really felt like this is a great field to be in, and we can really leverage these tools for either regenerative medicine or disease modeling for whichever aspect you're more interested in. So we've leveraged this technology quite a bit. We've shown that you can use these basic off-the-shelf plastics like polylactic acid rolls that are standard plastic that you can buy, that comes with like an off-the-shelf 3D printer that you could own in your own home today. versus ABS plastic, which comes from petroleum sources. So this is a non-renewable resource plastic, not biocompatible. The PLA is from natural corn reserves. It comes from cornstarch or sugarcane, and you can synthesize polylactic acid, and it's naturally biodegradable, and it's a good plastic alternate. So we showed that you can take these things, you can make scaffolds out of them that look like bone or any other type of tissue that you want, and you can use them at a high level to grow. In this case, we grew cartilage cells, or we grew spinal disc cells on them, and we showed that they maintain a high level of viability, and they produce the appropriate proteins, which means they maintain their identity and can behave more or less like that tissue in the body. So we showed that you can use these things, and this was going back almost 10 years now. We showed that you can use these things in high-level tissue engineering type studies. We got also interested in some other types of materials that can be useful in biofabrication to make tissue-like substances, but not only are they useful for making tissue-like constructs, some of these materials that we've worked with, we found that they are actually nanoporous, and they can be loaded with drugs or bioactive factors that can either inhibit, for example, tumor growth or promote bone regeneration. And we've gone on to publish several studies. This one here just shows that you can load it with a standard chemotherapeutic agent called doxorubicin, and depending on which type of material you use and which type of porosity it has, you can control the release of doxorubicin over time. Of course, the more porous it is, the more of the drug comes out, and the more it can disrupt, for example, prostate cancer cells here, or cell lines or patient-derived cells. The other interesting thing about these polymers that we've been working with is that we noticed when you look at high resolution scanning electron microscopy, this is what they print like, but when you wash them and part of the material comes out, you get these nice striated strands of polymer, and in this case, it's a polyurethane, and they look very collagenous. They look like collagen bundles that you would find in tissues such as ACL or tendons and things like that. What you see here, this is an electron micrograph I took from this study that just shows you what an actual ACL graph looks like. And the scale is not that far off from one another. So these are quite similar. So just what you're looking at here is that we've taken some of these scaffolds, we've 3D printed them, and then we've overlaid cells, which is an indicator of biofabrication. In this case, it's primary human ligament fibroblasts taken from patients in the operating room below us. And you can see this nice alignment of the cells across those striations of the scaffold that we printed. And this green versus red is just showing you green, all the living cells, and any red dot you see is a dead cell here. So we maintain a high level of viability, and the cells actually lay down extracellular matrix and behave the way you would expect them to behave. They start making collagen type 1, collagen type 3, they start making tenacin and things like that. So they're really behaving as though they're growing in an organism. Okay, of course, we can quantify. We did some other tests. Do you need to coat these materials with anything? Do you need to coat them before you put the cells on them? And the basic take-home message was you didn't really need to coat them with anything. The cells behave just well on them because of that biophysical characteristic. Let's shoot over a little bit to bioprinting, because that's what this talk was more about. Right, I just had to lay some foundations about what 3D printing is. So if we skip over to what's bioprinting, the difference is it is 3D printing, but it's 3D printing with living cells. Uses the same principles of additive manufacturing, except in this case, instead of running plastic or metal or ceramic through the device, you're actually running through it what's called a bioink, right? And a bioink is some kind of matrix. It's some kind of polymeric matrix. It could be naturally sourced. It could be synthetic. Usually a very popular one is collagen or gelatin or alginate. These are different kinds of gels that you can actually suspend cells in. And then you can either use a thermo device to droplet onto this surface that you're printing onto, or you can use some kind of extrusion device. So this is the most popular tool because it's the most inexpensive to make. So if you're buying a 3D printer or a bioprinter, this is usually the lowest cost. And very often it's either a piston-driven or a pneumatic-driven system. So air pressure is driving this out. So again, you can 3D print scaffolds, and you can position cells precisely where you want them. You can have one cell type in one area, another cell type in another area. Many of these 3D bioprinters come with multiple heads. So you can have 3 print heads, 6 print heads. Some of them have 12 print heads. You can have different materials with different mechanical properties. You can embed different growth factors in different tubes so that you get a gradient of growth factor effect. And most importantly, you can layer them and build geometric structures as you wish. Okay, so what are these bioinks? They're polymers. They're usually mixed with growth factors and or cells, and combined together, this is called a bioink. The most important thing that you have to worry about when you're selecting one of your bioinks to 3D print cells is the cytocompatibility of that bioink. So if you're making one yourself, for example, from a decellularized extracellular matrix that you've acquired from some kind of tissue, you have to make sure that it's cytocompatible and not killing your cells. You have to make sure that it has high precision printability, meaning that the gel holds its shape fidelity, right? So that's another important aspect, shape fidelity and precision printing. It has to be able to encapsulate your cells to protect them and also have attachment points for your cells if you want this to model an extracellular matrix. There should also be, you should also think about timely scaffold degradation, because if you wanted to use this scaffold that you're generating, for example, for regenerative medicine, that scaffold material needs to be degraded at the same time as new material is being produced in vivo. Okay, so what kind of materials can be used as a bioink for bioprinting and biofabrication? You have matrix-based bioinks, you have curing bioinks, you have sacrificial bioinks, and support bioinks. So what does that mean? Matrix bioinks are usually coming from materials like collagens and gelatins. Curing bioinks are bioinks that have been modified chemically with photoinitiators so that when you shine certain wavelength light on it, while you're printing, you can photocure and stiffen the hydrogel, because as you could imagine, certain hydrogels won't be able to be printed in layers too high because they'll collapse on themselves. Right? So if you use a photo curing agent, as you're building up that material, you can stiffen it by initiating the polymerization with a light source. Sacrificial bioinks, this is often called gel-in-gel printing. So this is when you have one type of hydrogel as a bath. Sometimes alginate is used, sometimes pluronic is used, sometimes gelatin is used. And what you end up doing is you have another hydrogel that's printed inside that gel, and then it can hold a nice shape. And then once your scaffold is cured, you can then wash away the sacrificial material and you're left with your construct that held its shape. Support bioinks means when you're printing with certain hydrogel cell mixes or some kind of matrix and cell mix, and you're overlaying that print over something like polylactic acid or polyurethane or polycaprolactone, which provides a much more rigid support for your construct. So those are some of the things: gelatin, alginate, scaffold-free slurries of just cells. That can be a matrix bioink. Igricure, calcium chloride. These are curing agents for curing bioinks. Pluronic, bioactive glasses, agaroses. Those are used as sacrificial materials. And of course, support materials can be polycaprolactone, polylactic acid, polylactic glycolic acid. These are all rigid support materials that will degrade very slowly over time. So here's some examples of some of the bioprinters we have in our lab. We use the BioX from Cell Inc. It's a 3-nozzle system. They also have a BioX6 that has 6 nozzles, and they also have the original Cell Inc, which has 2 nozzles. And this is another one that you can buy at quite a low cost from 3D Tissue Scribes. It's just a single nozzle machine, which you can see it's a regular 3D printer that's been modified to hold a syringe and press this using a gear screw to press this syringe down and print as you go. So this is a great way to get started if you're just getting interested in bioprinting. It won't break the bank. Units like this are quite a bit more expensive. But what are some things that we've done in the past couple of years? Well, we've been interested in modeling the bone microenvironment. We've developed our own bioink using a mixture of nanohydroxyapatite along with gelatin and alginate, different blends of gelatin and alginate. Gelatin is important because it provides protein binding sites for the cell surface proteins. And the alginate is important because you can rapidly cross-link that construct that you make using an ionic solution like calcium chloride. This is just the rheology that we've done to show that when you change a material, it changes the printability. So we changed this gelatin alginate gel by adding nanohydroxyapatite, which made it more liquefied and less viscous. That changed its bounce back from after it's being extruded out of the needle nozzle, which means it might not hold its shape as well. And one of the tricks that we did to get around that is we actually printed onto a cold surface, so that kept what happens when you put gelatin, when you make Jell-O, when you put it in the refrigerator, it stiffens more rapidly. So we used temperature to try to get around that, and it worked out a lot better for us when we printed on a cold surface. These are the kinds of constructs you can make. This is just an outer ring. We usually print 2 or 3 rings with different cells. And what this is just showing you is that this is a close-up, this is an electron micrograph close-up of alginate gelatin. And when you put the nanohydroxy inside it and you do a surface scan, you can clearly see all those little nanohydroxyapatite beads inside. If you look closely, when we put primary human osteoblasts inside there, you can see the osteoblasts nicely inside that gel. Of course, after you 3D bioprint something, you're pushing and squeezing cells through a small nozzle of a needle. You have to make sure that there's a good viability. So these are primary human osteoblasts that we get from organ donors, from spinal bodies. And you can see whether they're in the alginate gelatin without hydroxyapatite or the alginate gelatin with hydroxyapatite. You get a very high cell viability going several weeks out. You can take those tissues that you prepared using bioprinting and you can put them in specialized medium like osteogenic media, and we can drive here. What you're seeing is alizarin red S stain, which binds to calcium. So once you put with hydroxyapatite and an osteogenic media, those osteoblasts that are embedded in here make a lot of calcified matrix. That's great. What do we want to do with that? Well, we want to use this as a model for breast to bone metastasis, because we're interested in breast cancer metastasis to the spine. And what you can do is you can bioprint, photo labeled human breast cancer cells in that microenvironment that has primary human osteoblasts with the nanohydroxyapatite, and you can ask questions over time. Do the breast cancer cells migrate into this environment? Do osteoblasts migrate into the cancer environment? And what's happening at the molecular level in these cells? More importantly, we're interested in screening new therapeutics like different kinds of bisphosphonates. Can they disrupt this? We're interested in looking at in-house generated cements that can deliver drugs locally rather than systemically. Chemotherapy has terrible side effects, so if you can avoid those side effects, that would be great. But if we're building a biomedical device, we need a tool to study it before we go to an expensive animal study, and this serves as an excellent tool. So here's an example of this is just a patchwork of a lot of microscope images that were put together that shows where we can nicely print all these labeled, these are membrane labeled osteoblasts, and these are GFP expressing breast cancer cells with an empty gel ring in the middle. And this is a really nice model. We can resect it. We can let the margin, we can leave margins. We can put 3D printed scaffolds in here for testing drug delivery. We have cements with nanoparticles for drug delivery that we test in here. We have new types of drugs like anti-senescence drugs that we're testing. And we have a new project on testing cold atmospheric plasma for this. So this is how it works. You would have this and you can test some kind of biomaterial, see if it releases enough drug, for example, in this case, doxorubicin or zoledronate, and see if it can block that metastasis event of the breast cancer cells into that bone microenvironment. Here's an example of when we put a 3D printed scaffold. This is a nanoporous device, and we're just testing its release of doxorubicin against, for example, these are bone marrow derived MSCs. All the red dots are dead cells, all the green dots are live cells, and you can see that it's releasing enough drug to kill off half of the cells in this scaffold. Okay, we can quantify. We can then take our scaffold to quantify how many multicellular spheroids we have based on how much drug release we got. And again, this is showing you an example now of, I think this is supposed to be a video. It's not working. We're showing you how we have this plasma medicine device, and we programmed it on the CNC device to go into each well and deliver a dose of this, which looks like a little cigarette lighter here, but this is cold atmospheric plasma. and all of those little 3D bioprinted tumors in a bone microenvironment are in this dish. And we can test different dose responses there and get an idea of, will this be useful tool in vivo to block breast to bone metastasis? There's the video. So you can see it's being lit right now, and it's actually charged and treating this well. So what has one of our graduate students been doing? Well, they show you here, this is a live dead assay. That's the bioprint where the breast cancer cells are in the middle, and there's just stromal cells in the periphery. And she did a live dead assay after treating this directly with that plasma for, I think, 45 seconds. And you see that all the tumor cells are dying, but the stromal cells are more or less remaining alive. So you might ask, well, is that because the plasma is hitting right into the center? Well, she did the reverse experiment where she put the stromal cells in the middle and the tumor cells all on the outside, and did the live dead assay again. So all the green cells in the middle here are stromal cells, and all the red dots here are dead cancer cells. So this is really interesting way that we can use these 3D bioprinted or biofabricated tissues to screen new therapeutics. So let's fast forward a little bit. What's going on with regenerative medicine? Or has anybody made any leaps? We've all heard these stories about, bioprinting a heart, bioprinting a kidney. You may have seen a TED talk on this. Well, it's actually becoming a reality now. This is Dr. Larry Benassar up at, I think he's in Cornell in New York. And this is him about 11 years ago showing these bioprinted ears that he was making from patient scans. And he was then taking that scan, making a CAD model of a patient's ear, and then growing cartilage inside this ear. So about 3 years ago, he formed this company, 3D Biotherapeutics, with another professor, and they actually have finished just last year, at the end of last year, a phase 1/2A safety trial, putting these bioprinted ears with using cells out of the patient themselves to repopulate this ear. And they've completed a trial, and they're compiling the data right now. We'll see what's going to come out of that study. It looks very promising to me that this bioprinting an organ is becoming a reality. So we're getting really close to using these tools in regenerative medicine. Okay, so what about SkIN and biofabrication, right? This is the SkIN Canada group, and what can we learn from things that we're doing with these tools towards SkIN and biofabrication? So right away, when we look at SkIN, it's got 3 main layers. And even within, for example, the epidermis, it can be differentiated into multiple layers, right? You have different cell types in these layers, right? In the epidermis, you've got keratinocytes, you've got different types of keratinocytes, you've got basal membrane cells of the dermis interface. You have a lot of stem cells in here to regenerate the skin. And then, of course, in the dermis, you've got a lot of dermal fibroblasts that are supporting this. And then at the subcutaneous level, you got a lot of adipocytes and other types of stromal cells. Not to mention, you have blood vessels and immune cells here as well. You've got specialized follicular cells. So if you just look at this layering, it just makes sense, and it makes sense in my mind, and hopefully I can convey that message to you as well, that this looks like a very, very good target tissue for bioprinting, right? Because you can layer in different gels in a different, with a multi-head machine, you can make a 25 micron layer of gel that has just these cells. You can make a 30, 40 micron layer that has just these cells. I believe that this is only 0.1 to 0.2. You got up to 200 microns here in the epidermis, and the dermis can be anywhere from 2 to 10 millimeters, depending where it is in the body. Right? It can be very thin in the eyelid, can be very thick in regions on the foot and on your back. So you could imagine that you could layer all of these things in. You could even build in blood vessels to some kind of biofabricated or bioprinted skin model. What would that be useful for? Well, I did a little bit of research on this. This is not my field, but I did a little bit of research on this. So look at all these different kinds of skin disorders. Malignant melanoma. According to the Canadian Cancer Research Society, there's about 11,000 of these diagnosed per year, and it's increasing. This accounts, and of those, it leads to about 10% death. Okay, so maybe this would be a very good tool to 3D bioprint melanoma in a native microenvironment to screen new therapeutics or new surgical techniques. And maybe this would save time, money, and it would reduce the number of animals required to carry out a study prior to moving towards a clinical trial. Scleroderma and psoriasis, these are different autoimmune disorders that lead to different impacts, right? So scleroderma leads to overproduction of collagen and skin hardening. I found that there's in 2018, there's 2,000 existing cases in Ontario of this, which is very problematic. Psoriasis, which is often accompanied with inflammatory arthritis. You got 1 million Canadians affected. Burns worldwide, over 6 million people requiring grafting for burns. Well, there's a group out in France that is bioprinting skin as we speak. They have a patent on this. They have a specialized 3D bioprinter to make skin, and this is the dermal and epidermal layers. This is how they do it. They layer a bunch of fibroblasts, and on top of the layers of fibroblasts, they overlay keratinocytes. And this is what it looks like when they make it. You can see it looks really like a proper layer of skin. They've tested this in animals and shown that it integrates very well into an animal wound model. And they are actually in the process of initiating a clinical trial as we speak. So quick recap: 3D printing, bioprinting, useful tools for creating human tissues to model disease, create graft material for regenerative medicine. Bioprinted ears are being evaluated now following a phase 1/2A clinical trial. Bioprinted skin is going into clinical trials, and biofabricated skin, especially from patient-derived samples, could potentially be used for screening new therapies for inflammatory or cancer. So that's my 2 cents on how bioprinting can be leveraged for skin studies. Those are the people that were involved in generating all the data that I showed you from my lab. And thank you very much. I leave it to the next speaker.
Yeah, thank you, Derek. And we would like to take this opportunity to thank Dr. Derek Rosenspeak for an excellent presentation and for highlighting to us about the potential applications to skin bioprinting and bioengineering. So as I mentioned before, we'll call the questions for the panel discussion and move on to the next speaker, who is Dr. Rahul Gowri. I have known Rahul for a long time. He's a professor, assistant professor in the Department of Surgery, and his work is focused on the significance of using physiological oxygen tension in biomedical research, and in particular to 3D bioprinting. He will provide us an overview of the emerging methodologies in the field and their incorporation into research and how to leverage them in bioprinting and skin tissue engineering applications. He will highlight those aspects as well, along with his other musculoskeletal tissues. Gowri completed his PhD from McGill University and his MD from University of Calcutta. He completed an industrial internship with Johnson & Johnson in its division of spine division of Synthes, and he had already done the visiting fellowships at the University of Lund in Sweden and also EVO research in Switzerland. Dr. Gowri has also obtained several postdoc scholarships while during his training. He was trained at the University of Pennsylvania and also University of Toronto with the NSERC postdoctoral fellowship. Dr. Gowri does some kind of amalgamates his clinical and basic and translational expertise and his training in those areas to have a vibrant research program at McGill, focusing on novel applications of tissue engineering and in musculoskeletal diseases. His particular study that he would present to us will be focused on the role of oxygen tension in organ culture and animal models of diseases. Thank you, Dr. Gowri.
Thank you, Dr. Philip, for the generous introduction. Let me start sharing my slides. Okay, and dive into it. So I will keep the concept, the talk like concept heavy and data light, because Tariq is presenting after me and he's from the lab, so I will give him the opportunity to present the data that we're going to be talking about. And as a actual conceptual in practice thing that we are doing in our lab. So these are my disclosures. So tissue engineering. So I will give you a bit of a historical perspective as to what it means and where it lies in our day-to-day applications. And if we look into the use of tissue engineering and biomaterials, and before we delve into, Dr. Rosenzweig is a long-standing collaborator of mine, and it fits very well that he gave an overview about 3D bioprinting and creating those models as to how we can pre-screen, test therapies, make grafts. But then the fundamental question becomes that after we have printed them before implantation, how do we treat them? How do we culture them? And what are the concepts that we have to keep in mind before doing the graft implant so that there is a high success rate and there is no graft rejection? So as early as Neolithic period, sutures were being used to close wound healing and to close wounds and aid in healing. And ancient Egyptians were using linen and Europeans were using catgut as suture material. So knowingly, unknowingly, we kind of use materials that were closer to our own body or our own body structural molecules. Skin grafts were first employed by ancient Indians as early as 2500 BC. And the father of ancient Indian surgery, Shushruta, actually described in details how to take a gluteal flap along with fat and how to transplant that on face, cheeks, and areas where there are burns and there are mutilations because of wars. Sir Isaac Newton began to describe the body as a physiochemical machine, that it is in synergy between the physical forces, the biological forces, and the chemical reactions that happen every day. Abraham Tremblay in 18th century performed Hydra experiment. If you recall from our high school biology, if you cut one part of Hydra, it grows into the complete organism, meaning that we identified from very beginning that there are some organisms on this planet that have this high capacity of regenerating, whereas some others don't. So what can we do? What can we learn? What can be the applications that we can also regenerate our body parts to an acceptable degree and to get good outcomes? So Christian Ponder in mid-1800s also proposed that the cell environment is very important for tissue engineering. The cells or the tissue or the material that you graft at the site of injury or repair has to be synergistic with the local environment, else the physiology will not like it and it will throw it out. Because when we look in a lab, we look at these grafts as individual things growing in a petri dish, for example, but in a body, no tissue system is isolated. They all crosstalk, either via signaling molecules, local biomechanical forces, or the best messenger is our bloodstream, that it communicates between different systems. So tissue engineering triad is broadly divided into three axes: biomaterial scaffolds, then we have engineered tissue, we have growth factors, and we have cell source. So one, two, or all three of these axes can be used in successful tissue engineering and regenerative medicine applications, but they can stand alone, also be part of a very successful therapy. But when we put them together, either in twos or in all three together, this is to augment our tissue engineering efforts. And the aim is now targeting towards personalization, that how can we personalize these therapies, because we know that the concept of one size fits all is getting obsolete day by day, and with large data analytics and with the advent of new materials and large data sets that we have, we kind of know that how one body or one physiology is different from the other one. Over the years or decades, there has been refinement in the tissue engineering triad. So now, for example, the cell sources, we can engineer the cells by CRISPR technology or by inducing and creating iPSCs. From the engineering axis, we have these growth factor releasing biomaterials, we have smart biomaterials, we have biomaterials that can impart biomechanical forces effectively. And as Dr. Rosenzweig mentioned, the tissue architecture can also be mimicked. via self-assembling biomaterials or by 3D printing, doing additive manufacturing, either bioprinting or 3D printing. And we can also use decellularized organs as a scaffold. So further refinement is also happening, and we are going into very niche areas in each axis. So now to test out all these therapies, we have these cell sources that can be used as either sheets of cells or as organoids. And tissue architecture is also taken into account now. And we have this microarchitecture being printed in exquisite details in these tissue constructs. We are also playing with the niche properties like providing them with mRNA stimulation, miRNA stimulation, and with growth factors, transferring mitochondria, transferring DNA, RNA, and also biomechanical stimuli. And one of these niche properties is physiological oxygen tension, because that can also drive the tissue biology or tissue pathobiology. So I will just delve into the concept of physioxia. That what is physioxia? So physioxia or hypoxia, these are the terms that many people are not very clear with, and they're often confused, interchanged, and this is not true. So the term normoxia describes a normal physiological O2 levels in a tissue, and the organs and the context of in vitro and in vivo experimental systems. And it is used to describe the ambient O2 conditions to which tissue and culture cells are exposed to. Whereas hypoxia is a condition whereby tissue O2 levels are decreased either due to impaired blood supply or decreased blood O2 levels in the context of in vitro and ex vivo experimental system. Hypoxia is described at conditions set at less than 2% oxygen tension, whereas some studies have considered between 3 and 7 to be hypoxia as per experimental design. Physioxia, on the other hand, describes normal physiological concentrations within tissues and organs and is referred to as tissue normoxia. In general, O2 levels in each tissue organ or organs are determined by the amount of blood supply to that organ. And from a musculoskeletal point of view, we know that many of the elements in musculoskeletal physiology are highly vascular, like bone, muscles, skeletal muscles primarily. Whereas there are elements that are deprived of blood supply or they have limited blood supply, such as cartilage, intervertebral disc, ligaments, and tendons. And that's not detrimental. This is actually promoting their health. And if there is blood supply coming into those avascular or hypovascular tissue, it actually causes degeneration. And even in highly vascularized systems, for example, hepatic perisinusoidal regions, and liver is very highly vascular. The lowest oxygen levels in the body are found around perisinusoidal regions. So the body itself has redistributed the blood supply and oxygen tensions just to maintain that physiology and tissue level function. We have been exposed, or we are exposed from a very beginning in our life to these normoxic or physioxic levels of oxygen. For example, when doing in vitro assisted reproductive technologies, when we fuse an egg with a sperm, the mammalian tissue oxygen tensions are taken into account, which is between 2% to 8%. So even an embryo is exposed to low levels of oxygen until and unless the placental blood supply kicks in, and even the placental blood supply is giving them physioxia, not a big oxygen burst. And the latest guidelines that are published by the European Society of Human Reproduction actually recommended low O2 tension for embryo culture, because right now some of the labs are doing these assisted fertilizations in normoxic conditions, which I will describe later what normoxia in these conditions is. What about the role of relative hypoxia? So these people who live in high altitudes like high Andes and people in around Tibet and Nepal, it is known and proven that the high altitude environments, because of lesser oxygen tensions as compared to people living on the plains, they have better genetic and despite factoring in better genetics and lifestyle, they have a better cardiovascular health and disease development and a higher life expectancy. Acclimatization diminishes the risk of hypoxic conditioning, and there are genetic variants also found that help these people to survive and sustain and maintain their health in these high altitudes. So an overview is that our body is fairly highly dependent on the oxygen tensions we are exposed to, and within our body, it's like a compartment. Every compartment is unique, every compartment is personalized, and every compartment is uniquely predisposed to receive a certain amount of oxygen tension for their normal physiological function. So this is the composition of inhaled air. So we have the oxygen concentration of 21%. And this is the concentration of oxygen in exhaled air. So this is the amount that actually enters our body. And we exhale more carbon dioxide than we inhale, which is a known fact, which amounts to, when the air enters the lungs, we have 21% O2. When it's going to the heart, we only have 13% O2. Arterial blood is 9 to 10. When it reaches the venous system after feeding all the tissues, it's between 4 to 5%. Bone marrow, though it's highly vascular, has a oxygen tension of 2 to 7%. Whereas the musculoskeletal elements that I described before, like articular cartilage, only see 1 to 5% of O2 because there is a gradient. As far you are from the vascular bed, you become more and more hypoxic or more and more avascular and also hypoglycemic. And because the diffusion of nutrients and exclusion of excreta is through diffusion. Bone also, being highly vascular, only sees 5 to 10% oxygen tension. And what are we doing in our tissue culture conditions? So if we look here, we have around 18.6%. This is the input air when you feed the incubators with the gas, and after, mixing it with the CO2 that we provide and the water vapors that we have inside through our trough, if you all recall from our cell culture techniques that we have humidity levels. So it drops down to 18.6%, but it is still higher than what we are actually exposed to in our different tissue compartments. So what are the applications of oxygen tension in skin? So transcutaneous oxygen tension is a very commonly used technique. And this technique has been used to assess wound healing potential, screening for chronic vascular diseases, predicting the amputation level, prospective treatment for diabetic foot problem, because we know in diabetes, vasculature is compromised because of local endothelial damage, and to also predict the benefit of hyperbaric oxygen therapy. What about engineered skin grafts? So I'm not from the skin field, but my colleagues and what I read from different literature is that when you engineer a skin with the intent of grafting it, it takes around 15 days around 15 days for new vascularization. And this vascularization is accelerated if there is inosculation, meaning if the graft already has some elements of the vasculature coming in. And this is a very critical point in skin tissue engineering. And this angiogenesis can actually be driven by hypoxia, as we see in tumor biology and also in musculoskeletal biology. One of the master regulator is HIF1 alpha, which is induced by hypoxia. And we can see from this figure that how oxygen availability drives the HIF1 alpha upregulation, which in turn can upregulate angiogenesis and also increase the number of VEGF receptors to promote angiogenesis or drive them. So the idea is that when you have bioengineered, bioconstructed a skin graft or a skin organ on chip, is 20% or 18.6% oxygen a right method to be doing, or shall we expose them to a gradient before grafting them so that the cells in those tissue grafts are already primed toward angiogenesis? And as in my previous slide, as I showed, if these cells are already primed, when they actually see the graft bed, they have a higher chance of anastomosing or inosculation with the graft bed and have a lower chance of rejection. This is very significant in cases where diabetes and other metabolic disorders are to be encountered while doing a skin graft. Fibrosis and hypoxia is another application area. So we know that inflammation drives the cytokines, and hypoxia can also drive this process and can upregulate the fibrotic pathway, leading to the skin fibrosis and scarring of the tissue if there is not if there is an impairment in revascularization of the tissue or of the healing tissue in wound healing, for example. So at a cellular level, this hypoxia and all these oxygen tensions, what does it mean? So this is driven by mitochondria, as we all know, because we are mammals, we are eukaryotes, we are mammals, we are aerobes, but that's not true. We are facultative aerobes, meaning we can change our biomachinery into anaerobics if need comes to be. But that's not sustainable for long periods because our cell will eventually die. Same as in myocardial infarction, brain strokes. So in human, there can be over 100 or 1,000 mitochondria per cell. And if you look at the physiology, how it is distributed, RBCs don't have mitochondria. The reason for that is because they have a high oxygen carrying capacity. If they had mitochondria, by the time they deliver their oxygen cargo to the tissues, half of it will be utilized by the RBCs. So by evolutionary design, RBCs don't have a mitochondria so that they cannot use or chew up on the oxygen that they are carrying through hemoglobin. Human egg has the highest concentration of mitochondria, again an evolution design, because the egg, when fertilized, has to rapidly divide into the first embryo, gastrula, blastula stage. So when they're dividing, they have to also divide all the mitochondria. Mitochondrial DNA is circular, and it has 37 very critical genes, very small genome, but this is inherited from the mothers, and it is passing on to the offspring, and it has the potential to cause many congenital diseases. Mitochondrial dynamics. This organelle is the only organelle that has its own DNA and has the capacity to either fuse or fission. So when it fuses, it creates this less branched network, which I will show later on, to reduce the oxygen consumption and to switch over to anaerobic respiration, meaning there is a glycolysis happening in cytoplasm, and all those byproducts are used into oxidative phosphorylation in the mitochondria. So it disconnects its bioenergetics, whereas it fuses to create those massive chains and networks when oxygen is restored. This is very critical in cardiovascular biology and neurobiology. So this organelle can sense oxygen tension and modulate and try to preserve the tissue or the cells from injury and from eventual death. But at one time, when the threshold is reached, the cells goes into apoptosis, in which also mitochondria plays a very major role. There are many synthetic functions of mitochondria. As we can see here, it has a very pivotal role in fatty acid and cholesterol synthesis, in amino acid synthesis, which is also a backbone for creating collagens. We have nucleotide synthesis where mitochondria is involved with a series of intermediaries and enzymes. It can also aid in glucose and heme synthesis. So these are very critical processes controlled by mitochondria. And if we are not taking care of the oxygen tension in part of the cells, when they modulate, the downstream activities of the mitochondria are also hampered. So they modulate themselves in response to the oxygen tension, but in the end, these critical processes are also hampered or altered. So if we look into the realm of musculoskeletal disease and SkIN, it is also in the mitochondrial enzymes and its physiology is also involved in collagen maturation. Due to oxygen tension, it's very well known in wound healing literature that how oxygen bursts is required for the oxygenation of collagen chain, which I will not go into details, but it shows that if at a cellular level, we are not providing proper physioxia, it could be detrimental for our overall tissue engineering applications. So in our lab, we have done many stuff with mitochondria to assess their activity. I'm not going to go into detail because Tarek will be talking about them in more details. So there are many tools at your disposal, like mitochondrial dyes, voltage-dependent dyes. So you can either locate the mitochondria and see if there's drugs. For example, I was treating the cells with a biomolecule polyphosphates. So we have this tool available to us at a very high magnification. This is almost 100x image that you can localize these mitochondria, also look at the co-localization of these drugs and how they are relocating to these organelles. And this is a nuclear membrane where I can still see the drugs and is also relocating to the mitochondria. So all these probes are available. If you want to look at membrane potential voltage of mitochondria, there are voltage reporter dyes that are available based on your experimental needs. We are also delving into convoluting or deconvoluting these chains that I was describing, that when there is less oxygen, these mitochondria fuse. And when there is more oxygen, they make these branches, as you can see in these two figures. But how to quantify them? But this is very easily quantifiable if you have the right tools. So this is something also you can incorporate in your experimental plans and designs, because there are tools available at your disposal, and you can easily correlate that with a therapy or as a predictive model in various animal models or in vivo screening high throughput assays. We have also done confocal microscopy and looked at the mitochondrial activity post spinal cord injury in neurons. So there are different applications in different cell types. So this is not restricted to a certain organ culture, organ function. Electron microscopy is another application that you can use. Here we can easily see that this is a healthy mitochondria, and this is how an unhealthy mitochondria looks before death. There is widening of the cristae, widening of the membranes, and before the mitochondria itself kills and initiates mitophagy. And this organelle can actually eat itself up, meaning it just causes its own death to reduce its number. If it is not able to fuse and reduce its activity, it goes into mitophagy, as seen in many lysosomal storage disorders. This is a classical example that another tool that we can use in mitochondrial activity and physioxia, normoxia, hypoxia research. Proteomic analysis we are doing for many mitochondrial pathways to see that how these culture conditions are modulating these cells or these in ex vivo in vitro grown tissue constructs. There are different ways of inducing physioxia for research applications. At a chemical level, we can use cobalt chloride in the media. It's kind of a quote unquote metabolic poison. I don't want to call it metabolic poison. It's a oxidative poison of some kind that it can reduce the oxygen uptake and actually make the cells anaerobic, though there is enough oxygen present in the media. There are these Macintosh Fields jars used for bacterial cultures, and that can have complete anoxia or oxygen tensions that you want to have for a particular set of experiments. More refined is these small physioxia, hypoxia chambers, where you can induce physioxia or hypoxia. These are more controlled as compared to these jars. There is also these hypoxia incubator chambers. If you want to go a step further, if your facilities allow, there are tri-gas incubators that you can feed a premixed gas, put in the input numbers, and you can have the whole incubator having a very tight control on the amount of oxygen tension in the cultures. There are the dedicated, very advanced hypoxia workstations that has like zero variation in oxygen tensions, because when you're opening your culture under a BSC or a biological safety cabinet, changing the media, there is a time. It's approximately calculated to run 2 hours before that equilibrium is attained in the tissue cultures. So now if you have a time point for say qPCR, which is within 2 hours, are you seeing the right data or the cell still acclimatizing? So if the budget and the infrastructure permits, we can have these very fancy devices. So now you see that if you go back to qPCR or immunocytochemistry, how a small change in tension can off your data by like miles. So there are other applications for physioxia. There are oxygen-releasing smart biomaterials. You can coat them. So these smart biomaterials can actually deliver or capture oxygen from your blood supply or in tissue culture and provide the cells with the right amount. There is chemical modulation that we talked about. We can also do 3D gradient bioprinting that Dr. Rosenzweig talked about, because we know, for example, in tumors, the central core is hypoxic because there is no blood supply. So we can make that gradient. We can use physiologic chambers, and we can control the amount of sugar also in the culture media, which Tariq will talk more about, because that also modulates your mitochondrial activity. If you have the right or physioxic oxygen concentrations, the sugar can drive the cell to change their physiology. In our lab, we are currently using this system that we use the chambers and we flush these, and these are very sealed and isolated chambers, and they can be put in a regular incubator along with your control in what we call as normoxia, which is actually more oxygen that you're giving. So you can have these control experiments side by side to look at different oxygen tensions and how they affect these cells that are highly sensitive to oxygen tensions. So in conclusion, it is crucial to use physioxic culture conditions for tissue engineering and regenerative medicine applications. Regrafting, maturation of dermal grafts may increase the retention. With the advancements in technology and many imaging and bioanalytical tools available, Now, incorporating and tracking and quantifying physioxia is very, very simple. And many cues can actually be taken from research done in tissue that heavily rely on tightly controlled physioxic environments such as heart, neurons, tumor biology, oocytes, and musculoskeletal tissues. So you will see sometime that the clue or the hidden answer lies in related research areas. And this is where you can actually fine-tune your experiments. There is no such thing as a perfect experiment, but we can get to as near physioxic conditions as possible, gearing towards personalized medicine. Thank you.
Thank you, Dr. Gauri, for this wonderful talk or stimulating talk. I guess all of us use the oxygen tension, which we do not try to see the differences that we may experience if we are studying a signaling pathway. It could be very different in the presence of physioxy concentration as opposed to the normoxia that we use in our lab culturing system. So it's very much of an eye-opener for students to think about it, the particular system that they study. In this case, most of you are in the SkIN field. So what is the physiological concentration in the skin and its consequence of oxygen tension in the skin and its consequences on the endpoints that you study? Thank you, Dr. Gauri, for that very interesting talk. Now we will move on to Tarek Clayat. He is a PhD candidate in Dr. Gauri's lab. Tarek has a bachelor's degree in biology from the American University of Beirut in Lebanon. His doctoral research project focuses on utilizing biomechanical and bioengineering stimuli to fabricate implantable cell-seeded grafts for ligament tissue engineering and regeneration. Tarek has already received several prestigious scholarships. He has the provincial FRQS and FRQNT. He has also received scholarships from Oral Health Network here in Quebec. He has, I guess, several others that I don't have the list of. He is part of several scientific societies. He is the VP of Finance of our graduate program Student Society, the Surgical and Interventional Sciences graduate program at McGill. In addition, he holds the student training committee membership and positions in a Quebec cell network called TESEL. And he's also on the board of the student member of the Canadian Connective Tissue Society, or CCTS as we call it. Tarek will expand on the theme that Dr. Gauri has introduced, the critical role of oxygen that when you study tissue function. Tarek.
Thank you, Dr. Philip, for your introduction. I'll start by sharing my slides. Can you see my slides? Okay.
Yes.
Perfect. So today I'll be talking about one example of an application of physioxia and near physiological culture conditions here in tissue engineering, specifically ligament tissue engineering. So the ligamentogenic differentiation of human bone marrow-derived stromal cells using physiological oxygen tensions for tissue engineering applications. I have no conflicts of interest to disclose. So I'll start by introducing the anterior cruciate ligament. The anterior cruciate ligament is this ligament seen here. It's one of many soft tissue components of the knee joint, and it's key to the stability of the knee joints and ensuring the stable movement of that knee joint within the normal range of motion. And ACL injuries are some of the most common soft tissue orthopedic injuries. Now, tendons and ligaments are not only hypovascular, but also hypocellular, and their cells are mostly terminally differentiated. They're fibroblastic cells, and these cells have since they're terminally differentiated, they have a low proliferative potential. So these tissues, due to their hypovascularization, as well as their biomechanics and their cells, they have a low regeneration potential and they fail to properly heal after injury. Therefore, the current standard of care for ACL injury or ACL tear is replacing entirely the injured ACL or this ligament. It's called ACL reconstruction surgery. The ACL is removed and replaced by a tendinous autograft or allograft. However, this ACL reconstruction surgery, since we're replacing an ACL with a tendon, these are very similar. However, they still have differences. And this ACL reconstruction surgery, as a result, suffers high complication, failure, and revision rates due to multiple factors that we won't delve in due to the time. So there's clearly here an unmet clinical need for an alternative solution. And one alternative solution that is promising is tissue engineering. And as Dr. Gary already introduced tissue engineering and the triad of tissue engineering, if we want to recapitulate the special characteristic of the characteristics of the native tissue, and it's important to recapitulate these characteristics not only at the structural level, but because this structural level will actually dictate the function of these tissues and how functional they are, we should carefully choose the cell source, the biomaterials, which are ultimately the microarchitecture of the tissue or that we're trying to engineer, as well as the niche properties or the biochemical, biological, and biophysical cues that these cells and biomaterials are exposed to. So one of the important biochemical and biological cues here would be oxygen tension. And as Dr. Gary already mentioned, we have compartmentalization of oxygen tension within the different tissues of the body, with ligaments and tendons experiencing the least amount of oxygen, where it's reported in studies between 1 to 5%. And this is also true to the amount of glucose or the nutrient supply, as these tissues are hypovascular. So one might ask, how do these cells usually adapt, and how are these cells adapted to these environments within the body, for example, the fibroblast cells of the tendons and the ligaments? So Dr. Gary already introduced this organelle called the mitochondria and already went through all the important functions of the mitochondria that are found there. And one of the main also functions of the mitochondria that we mentioned is cell fate regulation, which would be key for stem cell differentiation other than energy metabolism and homeostasis. So what we hypothesized here is that that culturing bone marrow-derived mesenchymal stem cells under near physiological oxygen tensions and in combination with also near physiological glucose concentrations would enhance their differentiation into the ligamentogenic lineage for use in ligament tissue engineering. I'll start by a quick overview of our study design. So we have these human bone marrow-derived mesenchymal stem cells that we grow in differentiation media supplemented with 2 growth factors, TGF-beta3 and FGF2, which are known in the literature to drive the ligamentogenic differentiations of stem cells. And we conduct this differentiation or this experiment under 3 different oxygen tensions, 20% O2, mimicking atmospheric oxygen tension, and standard cell culture oxygen tension, versus 5% O2 and 2% O2, which are more physiological. And at the end, at the end of this 10-day incubation, we assess cell viability, metabolic activity, as well as the differentiation of these cells using gene expression to targeting or probing the expression of important ligament markers that I'll be talking about later. So this is the hypoxia device that we use. We have these hypoxia modular incubator chambers that can be sealed with the cell culture vessels inside, and then they're flushed with our hypoxia or in this case the 5% and 2% O2 concentrations that I mentioned, and then we can they're sealed and they're placed inside the incubator. So the first thing that we thought to determine is whether these cells are effectively experiencing hypoxia, and one of the things that we can do is look for the expression of a marker of this of hypoxia, which is hypoxia-inducible factor 1 alpha. It's a transcription factor that calculates hypoxia response in cells. It is always expressed, however, it's actually under normoxia or high oxygen levels. It's all signaled to by ubiquitination and then destroyed. So under hypoxia, this is not there and it's stabilized. That's why we can see it's a higher expression. And as you can see here in the immunofluorescence staining that we did by probing for HIF1 alpha, in the cells we have with lower oxygen tension, higher expression of HIF1 alpha, confirming that these cells are effectively experiencing hypoxia. Now, we then wanted to look at the cell viability. So whether this is actually feasible and we can sustain it for a long period of time, whether these cells are able to withstand these conditions. And as you can see, through this live dead assay, the cells that are glowing in green are live versus the cells that are glowing in red are dead, as Dr. Rosenzweig previously mentioned. We can see that 5% oxygen tension does not affect human bone marrow derived mesenchymal stem cells viability, with 2% slightly decreasing it. However, it's still in the range of 95%, and then we decided to look at whether these cells are also not only remaining viable, but still proliferating at this stage after 10 days of incubation under these near physiological oxygen tensions. So we looked at the doubling time of these cells, and what we observed is lower doubling time under lower glucose and oxygen tensions, meaning that these cells are actually having a higher doubling rate, or are proliferating more. Now, one of the main things that Dr. Gary talked about is the metabolic activity, and one of the things that we also looked for and assayed is metabolic activity. And one of the first assays that we did is Alamar Blue. Now, this assay actually is based on a reagent that is reduced using mitochondrial reductases. Therefore, it can tell us, it can give us some insight on the mitochondrial metabolic activity. And as you can see, we saw a decrease of this fluorescence or that signal of mitochondrial metabolic activity under lower oxygen tensions and lower glucose concentrations. However, when we wanted to understand, okay, so these cells are decreasing their mitochondrial metabolic activity, which we kind of expected, because we expect them under lower oxygen tension to shift maybe towards glycolysis or a more anaerobic process to produce ATP. And then we started doing more assays, such as the glucose uptake assay. And as you can see, we observed higher glucose uptake. And this was also expected and confirming our expectations, because since glycolysis or anaerobic respiration in that case is less effective than oxidative phosphorylation, which relies on oxygen, then we would need under anaerobic respiration more glucose to produce more ATP, or to maintain the levels of energy of the cells. So the last thing that I wanted to show you today, and that we probed for and that we screened for, is the expression of these key ligament markers to know whether this hypoxia, along with the lower glucose concentrations, is actually further driving the ligamentogenesis of these bone marrow-derived stem cells. So we looked at the gene expression of matrix proteins such as collagen type 1, collagen type 3, but also key proteins that are found also in the matrix, like tenascin C, a glycoprotein that regulates cell matrix interaction and matrix organization, as well as sclelaxis, which is the main transcription factor, the master regulator that actually regulates this phenotype, and the earliest known marker of the ligamentogenic and tenogenic lineage. And we observed higher expression, as you can see, of these markers under near physiological oxygen tensions and glucose concentrations. And based on our statistical data analysis, it looks like the 2% oxygen with 1 gram per liter glucose seems to be the optimal concentration, but this is to be further confirmed with more assays. So in conclusion, what I would say is that this study and these findings paved the way for these primed human bone marrow mesenchymal stem cells to be integrated into bio-inspired and bioengineered ligament grafts. These can be any hydrogels that were shown previously, or any of the 3D printed and bioprinted constructs that Dr. Rosenzweig talked about also in his presentation. And it would be also interesting to replicate these experiments in 2D, in 3D, when these cells are included in these constructs, and also mature these constructs in the near physiological culture conditions that we identified in the study. We're also starting experiments on human adipose tissue-derived stem cells, which are another source of stem cells that is used in ligament tissue engineering and that could be more readily available. And we're further optimizing our protocols for this application. Also, tissue engineering applications developed in this study can be used to bioengineer any other hypoxic tissue, such as cartilage and intervertebral discs, or for any other application of any tissue that one can imagine. Now, in terms of future directions, as Dr. Gary already hinted to, we want to further understand how these cells that I showed you are adapting to the new culture conditions and how they're shifting their metabolic activity. So we're doing more assays to look into other metabolites and whether they're maintaining their ATP production activity, their ATP production, as well as how they're regulating their mitochondrial network, which is a dynamic network. Because as Dr. Gary already mentioned and showed in his presentation, this mitochondrial network is and these dynamics vary and change during stem cell differentiation. So it would be key to understand how these things are changing in our differentiation experiments. And as I mentioned, I would like to, for my PhD project, to integrate these primed and differentiated cells in a bio-inspired scaffold that our collaborator, Dr. Showan Nazalt, developed in his lab, which is these dense aligned collagen gels to also not only mimic the physiological conditions in terms of oxygen and glucose, but also the matrix that these cells grow in to make these bioengineered ligament grafts. And at the end of this presentation, I would like to thank my supervisors as well as our collaborators and the funding agencies that make this research possible, specifically the Fonds de Recherche de Santé and Nature et Technologie du Québec for the doctoral scholarship I have from them. Thank you.
Thank you, Tarek, for a great presentation and showing us your excellent data. So now we have our fourth speaker, Brenda Fraser. Brenda, are you there? Are you? Yeah, so Brenda is there. Okay. So Brenda, I met Brenda just a couple of months ago, actually, in St. John's, Newfoundland, when I was attending the National Scleroderma Conference, and Brenda showed interest in interacting with the trainees and being part of SkIN Canada. So Brenda is here in her capacity as a patient representative and going to tell us about her lived experience with the skin disease. But more than that, I would like to highlight her other interests. She is a photographer trained as a crime scene photographer, as a police officer, and she has also traveled the world, traveling as an artistic roller skater, if I understand, representing Canada. So she on a competitive basis. And she currently, her interests involves sports photography and other event photography. So Brenda, go ahead, and we are looking forward to hearing your experience as a skin patient. Thank you.
Thank you so much, Dr. Phillip. And yes, I'm from Hamilton and had the pleasure of meeting Dr. Phillip recently at the conference. And I've been asked just to share some of my experiences living with a disease of the skin and hopefully being able to provide a bit of a human element to your research so you can see that, yes, your research really does matter. And we're very thankful for everything that you're doing on your side of things. I'm first going to address the tightening of my skin, which is a part of scleroderma. And it not only affects my appearance, on a positive note, I'm hoping maybe my wrinkles will be less as I age as the normal person. However, it does cause a shrinking of the mouth and tightening around the mouth. So my ability to enunciate is a little different than it used to be. I've given thousands of presentations in my previous careers. So I apologize if my cadence is a little slow. I am kind of basing this on the fact that Siri always gets what I'm saying wrong in the car. So clearly I'm not speaking as well as I used to, but bear with me on that. And I'm not going to take too much time here. So I'm 5 years into my sclerodermic diagnosis in November, about 5.5 years from symptom onset. I'm 59 years old, turning 60 in January. No medications up until 55 when all of this started. I was very healthy, but blindsided with this life-altering disease that I'd never heard of before. So as mentioned, I've always been healthy. I've represented Canada in sports. I've maintained a healthy lifestyle, not radical, but healthier than most people. 36 years professional experience. So I worked in aviation at the airport as an airport agent, 25 years as a police officer, and then I returned to aviation 7.5 years ago as a flight attendant and a flight attendant supervisor and instructor. And I'll get how this disease has impacted my ability and not being able to do a lot of things anymore. So in April 2019, I first noticed an experience of Raynaud's, which I had never heard of. I came out of a night flight in the morning, my hands turned white, but I really didn't think nothing of it. It was just a little bit odd. May or June 2019, I noticed running my hands under hot water. I had a painful sensation, like irritation under my skin is how I described it, but also my fingers were swollen and I couldn't take my rings off to shower, which was unusual for me. I always had skinny fingers, never had swelling, but thought, well, it's May, June, it's kind of getting humid. Maybe I'm aging, maybe swelling is a part of aging. So I just kind of wrote that off. But then a more severe Raynaud's experience in August, walking into the grocery store, my hands turned blue. So again, not knowing what Raynaud's was, that's when I went to the doctor, we started testing, and I received the sclerodermic, the diagnosis in November of 2019. At that time, the symptoms were relatively mild, other than the skin, the swelling. Over a few months, though, I started to notice pitting in the skin and losing the dexterity of my hands. So that's kind of how it started to affect me a little bit more. Joint skin, like skin tightening of my arms, unable to raise my hands above my head because of tightening of the skin around my trunk and my arms is what I kind of noticed at the start. Like it was difficult to just shampooing my hair. Pigment changes in my skin. So on my hands, especially white patches on my skin, which again, it's just now I accept it, as well as the pitting and the calcinosis has become more severe over the number of years, probably over the last 2 years, I would say, that it gets a little bit more difficult. Tightening of my arms and my shins, I feel, as well as sometimes itchy skin, which is part of this, and red spots on my face as well. The sensitivity to warm water continues, and even it's progressed to the sensitivity to like what I would describe as rough fabrics. So wearing denims or even like socks, the seams will affect my feet because I have some of the calcinosis on my balls of my feet as well. I have to like buy a purse that doesn't have a metal zipper because just my hands touching the metal zipper causes irritation or could also tear the skin, which then could lead to infection, right? So it's kind of a lot of management of things that I never thought I would ever have to be managing. Digital ulcers is a problem. They can become extremely painful and take time to heal, sometimes requiring antibiotics to do so, but also just the sensitivity of the fingertips and just typing. So everyday tasks typing. The thickening of my skin and my forearms over the past couple of years, but noticeably the last year, it's making it more difficult for them to take blood. I go for blood work every month, so it's more difficult. They might be having to move to the top of my hands to be able to find a vein. So that's kind of what I'm experiencing as well. As far as like these medical changes, but just lifestyle changes. Like I find I've had to replace my cutlery because it was more of a squared knife and fork as opposed to rounded edges, plates that are lighter, mugs that are not like nice heavy stone mugs that are lovely to look at, but something that's got a rounded handle that just doesn't irritate my hands. It's just really bizarre the things that I've noticed that I've had to do to deal with this. And then as far as photography, so sports photography is what I got into after policing, but I don't have the dexterity to be that quick with my camera anymore. So I am switching to other things, astrophotography, landscape, and just more of a hobby and less of a business now, unfortunately. Being able to run, I don't know that I'll ever run again. Like I ran a marathon 6 years ago. I now have a accessible parking pass and I can barely walk 2 kilometers without breaking, having to stop kind of thing. That's not really skin related, but it's overall effect from the entire disease. playing ball because of the limited mobility in my shoulders. And I do go to physio, yoga, and things like that to try to maintain the range of motion that I have to keep the SkIN supple as best I can as well. And yeah, I'm just very thankful for this opportunity to speak with everybody here. And I just wanted to say that I'm so thankful, and it's reassuring to know that research is going on in the background to hopefully be able to better understand skin disease and to be able to help the people that are living with it. So thank you for this opportunity.
Thank you, Brenda, for sharing with us. We truly appreciate you. It's not easy to share these things, but you have you shared this with us, and we truly appreciate that help. And the trainees are very always are interested in hearing about the diseases that they work on. In our lab, we do work with scleroderma samples, and so it's very important for us.
Yeah, I might be part of those samples because I have been part of the other ongoing research out of Hamilton too. So thank you.
Thank you. Thank you. It's a rare disease, so it's not easy to obtain samples, but we do get, a few of them, few samples from the rheumatologists. Yeah. Okay. Yeah. So now we will move on to the panel discussion where we'll have the opportunity to ask all 4 speakers questions. So I guess please feel free to unmute yourself and ask any questions that come to mind. And thank you for participating actively. We encourage all the trainees to be interactive and participate in this discussion. Thank you. So who would want to start? Yeah, Valentin.
Valentin, good afternoon, everyone. Thank you so much for this beautiful and straightforward mini conference, in fact. I have a question for Derek. So one, it's related how he gets vascularization. So when he's doing 3D printed constructs with redoxapatite for bone, did he get cells? So first, what kind of osteoblasts he's working with, and how did he really get a true bio osteointegration of the graft, or he's not yet there in terms of direct application in clinics?
The question is directed toward Dr. Gauri or Dr.
Derek.
Derek, yeah, okay, yeah.
About
Dr.
yeah.
Yeah.
Okay, so let me get it straight. So the first part of your question is what was the source of the osteoblast?
Yeah, what kind of cells osteoblasts are you using?
Okay, and then the second part of your question is how are they integrated within the construct that we make?
Yeah, and if you are there, in fact, just an experimental research playing around with materials, or you get insights in clinics, like also integration in patients, at least.
Right. Okay. So the first answer to your question is they're coming directly from vertebral bodies of spine, so directly out of bone from organ donors. Okay, so we get them right out of the cortical bone mostly. Sometimes we get some spongy bone, and we have to process this using a special methodology, and it takes on the order of 2 weeks or so to get those cells out of a series of washes, enzymatic digestions, and then plating of actual little pieces of broken bones to getting the cells out.
Yeah. So you have how to say, we have validated procedure to produce these cells, actually.
Precisely.
Yeah.
Right. So those are published methodologies that we use, right?
Yeah.
The second part of your question is a little bit trickier to answer because we don't put those samples into an animal or into a bone or anything like that.
Yeah, not Yeah, not yet. Probably there will be at
So
a later
the
date.
idea behind those samples that have osteoblasts and nanohydroxyapatite is we're using it just to model tumor microenvironment.
Ah, okay, okay, modeling.
So imagine
Yes.
like a
Mhm.
96 well dish with different concentrations of drugs being tested, but in every single well of the 96 well dish, we've precisely placed the same number of osteoblasts, the same number of tumor cells from the same patient, whether we're getting it from here or there. So they can be highly reproducible in every well. So we have some sort of high throughput in a reproducible manner.
Yes. Yeah,
So the
thank you very much.
intention, yeah, so the
I
intention
understand.
there is not to implant,
Understood.
it's to use it for therapeutic screening.
Great. Thank you so much, Derek.
Okay, other questions? I can't see anything in the chat yet. Not yet, yeah. Okay, anyone else like to unmute and ask questions? Meanwhile, perhaps I can ask directed towards Tarek and Dr. Gowri. In terms of choosing your stem cell, how do you choose whether it is BMSC versus adipose derived? How do you make that decision?
Tarek, you want to take this or you take it and I help you? Because for the trainees.
Do you want to start? I will continue. Do you want me to start? I continue.
Why don't you continue and I polish you? As a
Okay.
patient supervisor from the outside, I'll just give you
Yeah.
support.
Okay. Yeah. So, as Dr. Gary and I mentioned in our presentation, there are multiple cell sources that we can think of. There's a lot of research that, for example, in the field of ligament tissue engineering and tendon tissue engineering, on different types of cell sources and which respond better to the growth factors, non-growth factors, or the mechanical stimuli or the bioenergetic stimuli. So, what I would say about the choice of the cell source, how do we choose if it's bone marrow or adipose derived? I would say adipose derived would be a better source that if we want to go to clinic, because it's easier to get. But then we would have to validate its ligamentization potential, which in the literature has been shown to be very variable depending on which area of or what is the source of fat where we're taking the adipose derived stem cells. Bone marrow has been used a lot and has been showing promising results in the literature. Another cell source that I also use is primary human ACL cells, so differentiated cells, and we use them in the lab. So these are usually parts of the tissues that are discarded because these ACLs are injured. But what we're trying to do is to maintain their phenotype in vitro so that we can harness these cells for ligament tissue engineering applications.
Yeah, so that,
So
yeah,
to
that's
add on to that is that, as Tarek mentioned, that we try to get the source, which is as easy as possible, because we are heading towards personalized medicine. The idea being that, or we know with large genomic data analysis that there are many, many differences, and these cells, when implanted, might not like my body environment, for example. So these adipocytes are very easily accessible, and many people undergo plastic surgery and this is discarded. So rather than discarding them, you can store them, because many of these plastic surgery clinics, they offer that service. Then instead of discarding your fat tissue, they will process the stem cells out and cryofreeze as same as your embryonic stem cells that we do with different companies when the child is born. Now, we didn't have that option, but I don't know if it works or not. Maybe in the future it will. So now you can still bank your cells and you can use it as an autograft. But then the idea here is that Tarek mentioned that these stem cells are very different from bone marrow derived stem cells. For example, their mitochondria is entirely different. Even within your body, where you get the fat cells from, it highly varies. We have brown fat, we have white fat, we have yellow fat, and then their mitochondria is called mega mitochondria because they're very big, because they are used to like more energy consumption and making more fat. So when we have this viable source, we think from a tissue engineering perspective, oh, easily accessible, minimally invasive. But if you don't grow them under right culture conditions, they might not behave properly. And that's why we see in literature that adipose-derived stem cells have shown marginal benefits, though they are a very good cell source. But then researchers, they fail to understand that you're putting a stem cell, comparing it to bone marrow stem cell with an entirely different microenvironment. And these stem cells, though you can argue saying that, oh, it's the same amount of oxygen tension, But you look at the mitochondria, there is three times as size as compared to bone marrow cells mitochondria. So will they behave the same way? Will they fuse? And what are they doing that might cause cell death? So these are the basic cues that we have to keep in mind before we go
Yeah.
further on and make those bioinks and make those fancy grafts. What do we put in? So that was our source.
One has to streamline the cocktails used for
Yep.
differentiation as well. So I think Ken has a question. Ken, go ahead.
Yes, I have a question for Derek, not Tarek. So Derek mentioned, you know, for the 3D bioprinting, the idea of putting printing endothelial cells in addition to other cells. So I'm just curious if that's ever been done, and what happens to the endothelial cells? Do they actually start forming vessels and take on like a physiological like structure, or do they just kind of you know, sit there in a monolayer, or what happens?
Yeah, it's a good question. So there's a few labs that are specializing in this. One is Burdick's lab. He's out in Colorado now. He went from Penn to Colorado. And there's Osbolat's lab in I think he's in Penn State. And they have various techniques with this gel-in-gel printing using sacrificial materials. So they'll print like a tissue, like a bulk tissue, and sort of like a full thickness hydrogel, maybe 1 millimeter thick, something like this, maybe 2 millimeters thick. There's also, sorry, I left out. It's Jennifer. I can't remember her name. She's at Harvard, MIT. They're also doing this, where they actually are printing these sacrificial channels. So as they flush out that sacrificial material that leaves a cylinder, they have a trick where they can do chemistry on that gel, which will have specific binding sites for endothelial cells, vascular endothelial cells. And they can actually populate these channels using this technique and show that several weeks out, they can have like a culture media perfusing through those channels separately than the rest of what the tissue sits in. And they can maintain a high level of viability of vascular channels. There are other people that are working on like spontaneous vascularization with co-culture, various types of co-cultures, but I think that it doesn't work as rapidly or as well. So there's other biofabrication techniques, as you all may well know from the LOX, for biofabricating vascular networks using sheets of cell materials and rolling them and then embedding those in other tissue grafts. So there's multiple ways to do it. There's also microfluidic devices to do this as well, which kind of goes beyond the realm of bioprinting, but nonetheless, yeah, there are ways to do it. We haven't been doing that too much. We've been just relying on direct co-culture using very thin bioprinted, like 2 layers thick, under 0.5 millimeter, so that oxygen tensions, like Dr. Gowrie was saying, don't play such a big role in the little tissues that we're growing. But it could become problematic if you go thicker and thicker and more complex, where you would need such things as gradients or various oxygen tensions in the system altogether.
Okay, yeah, I think that'd be important if you're making a SkIN model to mimic that situation. I think that would be the best strategy for sure.
There are methodologies for it, yeah.
All right, thanks.
Okay, thank you, Derek. Yeah, thank you. Meanwhile, while some others trainees think about their questions, I'm hoping that people would be able to be more interactive. Okay, I have a question for Brenda. Brenda, could you share with us your experience with the medical system here, your access and all related stuff?
Yes, I've been overwhelmed with the amount of support. So I'm in Hamilton, and I was dealing with Dr. Maggie Larche. She was my first rheumatologist that was me at the start of this, out of St. Joe's in Hamilton. Fantastic team. Like I said, I had no medication up to 55, and now I have a team of doctors and medication that gets delivered to my house. However, having said that, it's all helped me feel better, as well as extremely supported, and as well as my own family doctor. So together, everything is, I've felt very well supported. in the care that I've received. And like I said, the conference was fantastic. And then finding out that there's research going on behind the scenes. I've lost my ability to maintain certifications as a flight attendant. Therefore, I've lost my job as a flight attendant manager or instructor. I can't do that anymore. But being able to articulate the reasons of my disease and as far as medical accommodations through the medical field, the partnership has been fantastic. So I'm very impressed with it, that even though I have this crummy disease to deal with, I feel that I might not see the benefits, but it's great to know all this research is happening. And it's great to be able to meet you all as well, and to be heard, and to realize I'm not alone, that there's people out there to help and that are working on things. So again, I very much appreciate that.
Yeah, thank you for sharing with us that positive experience that you had with the medical system. Some people talked about the difficulty sometimes in diagnosis and all that, but you didn't experience that. That's very positive. Thank you.
Yeah, if I can just add, the one thing I have experienced is there's one medication just for the Raynaud's for circulation that I'm too young, so Ontario will approve it for scleroderma. However, I'm not 65 yet, and I have a medical plan, but my medical plan doesn't cover it. So that's one fight that I have to fight. But otherwise, like I said, I felt extremely supported and confident in my physicians.
Yeah. Championship is so important, and it's also helped us researchers very much. When you go to Ottawa and champion your own cause, it also indirectly benefits research. The politicians here only whoever has the loudest voices and votes.
Yes, well, the Hamilton, the scleroderma group, we're going to Toronto to Queen's Park anyway, the first week in November.
Yeah, thank you. Yeah, okay, there is a question, Adele.
Yes, I have a question for Dr. Gari. So like I study basically like skin fibrosis, scleroderma, and inflammation. And like I mostly focus on the NF-kappa B signaling pathway. And I saw in one of your slides you had it mentioned with hypoxia. So I was just wondering if hypoxia can cause like an effect on the NF-kappa B signaling pathway. Because I know that in scleroderma, I was reading on it that since the tissue has like less, like it's more stiff, there's less oxygenation. So there is hypoxia in scleroderma. So I was wondering if that would be something useful.
Thanks for the question. If you look at the literature, HIF1 alpha has been known to induce NF-kappa beta pathway or modulate its activity. So it's like a feedback loop that initially it's protective because we need that upregulation because NF-kappa beta is also involved in MMP production, for example, which is needed for remodeling of the newly laid down matrix. So at one threshold, it's good to have that kickback that it is actually bringing up those pathways. But after a certain level, it becomes detrimental because it doesn't switch off. So for example, when you're mentioning about different substrate strengths and stiffness, this makes the cells kind of undergo a sustained hypoxia from physioxia, and that might trigger and further that effect. So that's why even the material that are chosen, like for example, Dr. Rosenweig works in different materials with different stiffnesses. They're chosen which are more physiological because we know that even the substrate hardness can reprogram the cells or make them miscommunicate the signals going to them. So that's something you should definitely incorporate and look into your experiments as a culture condition, that modulating that as in patient samples and versus a healthy control. How does it differ? And what is the effect? And if you block that, will it reverse that process? Meaning that in addition to drug therapy, maybe hyperbolic oxygen therapy that is currently used might actually be also beneficial.
Thank you.
Thank you so much. I think if we have no further questions, we have gone over time, but it was very stimulating. Let us thank all the 4 speakers for talking to us and giving us a highly stimulating talks, especially Brenda, who who was willing to come and talk to us, boring scientists, but she has really shared with us her own experience. lived experience with the disease, which was all very stimulating to all the trainees. I hope you all will go back to your labs more stimulated and more, you know how it can affect patients in real life. So anything that we are all laying bricks, and one day something will come out of it with everybody's laying those bricks. Discoveries come from maybe only once in years, but that they do come. So get encouraged and go for it. Thank you so much.
Thank you very much.
Okay, so
Thank you.
I'll end. So Ken, you will end the conference now? Yeah.
Yes, I will. Thanks,
Thank you.
everybody, for joining. We'll see you at the next
Thank you.
webinar, which we will
Thank you.
advertise when it's
Next year, it will be next year. Thank you.
Thank you
Thanks
very
for
much.
having us.
Much
Thank you.
appreciated.
Thank you, everyone.
Bye-bye.
Bye.
Thank you.
Bye-bye.